Well, hello everyone. This is Varangi Mahadeshwar, and today we are going to talk about enzymes and proteins that are involved in the DNA replication process. So, what exactly is DNA replication? DNA replication is a process where the cell makes an exact copy of its DNA, ensuring that the new cell has the same genetic information as its cell. Now, this is crucial for the growth and development and the repair in living organism. But how does this intricate process happen so accurately and efficiently? That's where the enzymes come into the picture, each playing specific role to ensure that the job is done right. Let's break down the key enzymes involved in the process of DNA replication. So, the crude enzymes that are present in both prokaryotic and eukaryotic DNA replication are as follow: topoisomerases, helicases, primase, single-stranded DNA binding protein, also called as SSB protein, DNA polymerases, and ligases. They all together form the DNA replication complex. Let's first start about the topoisomerases. Imagine that you are trying to untangle a twisted rope. Topoisomerases are like those hands that prevent the rope, that is a DNA, from becoming too twisted or supercoiled. They do it by creating temporary breaks in the DNA strands to reveal the tension, then rejoin them. As the name suggests, they come under the class of isomerases, and their EC number is seen on the screen. Topoisomerases have several key structural components, starting off with catalytic core. Catalytic core contains the active site for cleaving and rejoining of the DNA strands, including the conserved amino acid residues and the metal ions. DNA binding domain, these domain recognizes and interacts with the DNA, facilitating cleavage and rejoining processes. Then there are the gate domains. These undergo conformational changes to open and close the active site, allowing the DNA to access it. There's a linker region that connects these different domains and coordinating movements and interacting the catalysis activity. There is also the presence of dimerization interface. These enables the formation of active enzyme complexes by facilitating dimerization. Regulatory domain. These modulate activity in response to the cellular signals or protein interaction affecting the DNA binding, catalysis, and localization. The two pictures here are of the structure topoisomerase 1 and topoisomerase 2, whose function we'll see in the further slide. Functions of DNA topoisomerases. The main function is to prevent superhelical tension, to mitigate the superhelical tension generated ahead of the replication fork by transiently cleaving and rejoining the DNA strand. Helix relaxation. The type 1 topoisomerase induces transient single strand breaks, facilitating relaxation of the negative supercoils and reducing the torsional stress, whereas the type 2 topoisomerase induces transient double strand breaks, altering the linking number in the increment of 2 and efficiently managing the positive and the negative supercoils. It also helps in the progression of replication fork by resolving supercoiling Topoisomerase enables the continuous progression of the replication fork, ensuring that the DNA polymerase can traverse the template strand unimpeded. Genomic stability maintenance is also done by topoisomerase, that they maintain the structural integrity of the genome by preventing topological stress, thereby reducing the risk of replication associated damages and ensuring high fidelity replication. One of the special functions in prokaryotes done by topoisomerases is utilization of ATP to introduce negative supercoils, which is critical for the compaction and functional regulation of bacterial chromosome. This is done by the type 2 topoisomerase called DNA Ligase. Now, let us look at the helicases. Helicase can also be called as molecular scissors, as they cut through the hydrogen bonds holding the DNA strands together. This separation creates a single stranded DNA template needed for the replication. These helicases come under the class of hydrolases as they hydrolyze the bonds present in the between the nucleotides. Now, the EC number of the helicases is given onto the screen. Now, helicases have several important structural components, starting off with the N-terminal domain, which binds to the DNA using helix-turn helix motif for the stabilization, and the C-terminal domain, which binds to ATP and has an ATPase activity to provide energy for function. There is also three motor domain which unwinds the DNA by breaking the bonds of the nucleotide. There is a rec A fold, which is because helicases have single NTP binding sites and have distinct polynucleotide binding sites that are allosterically linked. Now there is subdomain 1A and 2A, which contains a REC-A-like fold and is conserved helicase motifs, forming a groove for the single stranded nucleic acid binding. There is also a ring hexamer structure, which is a subunit interface, includes the conserved helicase motif and NTP binding sites. A critical arginine residue facilitates conformational changes. The central channel accommodation single strands of DNA or RNA and adopts multiple conformation. Talking about the function, the major function is unwinding of the DNA. Helicase separates the double strands at the replication fork by breaking hydrogen bonds, creating single strand templates. Then there is formation of replication fork, which creates a forked structure essential for the DNA polymerase to synthesize a new strand. Then there is loading onto the DNA. Helicases is loaded onto the single stranded DNA to initiate replication with working proteins like DNA B interaction with other proteins. It forms a primosome with DNA G primase, which synthesizes RNA primer for the polymerase. Now, chromosomes here is mainly formed when the DNA, that is, when the helicase and the DNA primase are in close proximity with each other, which makes sure that the position from where the replication is going to start is accurate. Then the formation of Okazaki fragments. is also it's where also the DNA helicases contribute, and also they are seen to function in the recombination and repair mechanisms. So here we can clearly see that how helicases are cleaving the bond that is present between the nucleotides uh, of the double-stranded DNA. Moving on towards DNA primases. Primases lay down short RNA primers on the single stranded DNA, providing a start point for the DNA polymerases. It's like a primer coat you put on a wall before painting it. Now, these DNA primases come under the class of transferases, and their EC number is given onto the screen. Primases have an important structural feature that enables its function. So, starting off with a cationic cluster that it has, this cationic cluster is like a magnet that holds onto the single stranded DNA using electrostatic forces, keeping it steady while the primases work. Then they have the zinc finger motif, which keeps the whole structure stable and helps to grab the right building material, which we can completely see in the attached photo, where the zinc binding domain acts like an attachment, which will make sure that wherever you are getting attached is at the right place. Then we have the RNAP motif, which initiates the synthesis of RNA primers, just like turning the key starts a car. Then there is also presence of magnesium binding protein. These sites are like the engine oils that keep all the parts running smoothly. Magnesium ions are essential for the catalytic activity of primases. Here in the above picture, we can see how the RNAP domain and the zinc binding domain work together in the RNA primase. Then we have the N-terminal domain and the C-terminal domain. N-terminal domain acts like a blueprint of the construction project, helping primates identify where to start the building RNA primer on the DNA template. The the C-terminal is the region which is vital for the formation of chromosomes. So, this chromosomes we talked about in the last slides of helicases. So, the C terminal attaches with the helicases and they form chromosomes. So, the here the C terminal has importance in formation of chromosomes. Then we have a hinge region which allows the primases to adapt and interact with other replication proteins. Then there is a presence of alpha helical tail. This tail aids in the interaction with helicase, ensuring the primase is correctly positioned for the efficient work. In the structure, we can see the zinc, the N terminal, the C terminal. Now let's talk about the function that DNA primase does. So, firstly is RNA primer synthesis. They create short RNA primers on the single stranded DNA, providing from the 3' hydroxyl group needed for the DNA polymerase. Then it initiates the replication process. Primase is essential for starting DNA replication. At the origin of the replication, it starts the initial RNA primer necessary for the DNA replication to commence. Then the RNA primers are the main uh, region for the Okazaki fragment formation. So, on the lagging strand, the DNA synthesis occurred discontinuously in short segments called as Okazaki fragments. Now, these primases synthesize multiple RNA primers along the lagging strand, allowing the DNA to create these fragments. Thinking of this process, it's like laying down tracks of the train that's being assembled in a segment. Further, talking about the starting of the stalled forks, in sometimes the replication forks get stalled due to various reasons like DNA damage and etc. Primases help to restart these DNA replication forks which are stalled. Then we have the interaction with helicases, where we talk about the chromosomes that are formed with the help of the helicase and the RNA primase, which we can see in the uh, diagram that I have added, where the DNA G primase is binded or is in close interaction with DNA B helicases, forming RNA primers, which ensures proper function. Then there is template binding. So the zinc ribbon motif, which is present in the prokaryotic primases, aids in the binding to the template DNA. This ensures that the primers are synthesized accurately and at the correct location on the DNA templates, like a tailor measuring or cutting a fabric precisely to create a perfect fit. Then, talking about, we have the single stranded DNA proteins, also called as the SSB protein. Okay, so moving on to the next, that is the single stranded DNA binding protein, or also called as SSB. These proteins play crucial role in stabilizing the single stranded DNA during the replication. Now, these SSB can be compared to a protective plastic cover used to keep the documents safe, just as these covers prevent the papers from becoming damaged, wrinkled, or stained. Similarly, the SSB binds to the single stranded DNA during replication to stabilize it, prevent it from any damage, forming secondary structures. Uh, and a lot of functions it has, which we'll see further. Talking about the structure, it has an homotetrameric form, which is composed of four identical subunits. That is, SSB forms a stable complex that binds to a single stranded DNA. Then it has N-terminal domain and C-terminal domain, where the N-terminal domain is like an adhesive side uh, of a Velcro, ensuring strong attachment to the DNA backbone. And the C-terminal domain is like which is rich in negatively charged amino acid, and it plays an important role in protein stability and DNA interactions. So along with this, it also have uh, DNA binding domain, which is located like approximately 115 amino acids from the N terminus. This domain is essential for the binding of the single stranded DNA, like the loops on the Velcro that catches onto the hooks. Um, yeah. Along with that, it has two binding modes, that is low salt concentration and high salt concentration. And the subunits it has are alpha helix, beta sheets, and random coils. The structure of the SSB can be clearly seen in the um adjacent images. Moving on to SSBP's P's function, they have several important functions. Firstly, is the stabilization of the single-stranded DNA. That is, single-stranded DNA binding protein prevents the single-stranded DNA from reannealing or forming any secondary structures like the hairpin loops, which it can get into because of the uh, nucleotides that are present onto the strands. Then it also helps from the protection from the degradation by binding to the ssDNA. The single stranded binding protein protects it from the degradation from the nucleic acids by being a protective cover or shield. Then there is also the facilitation of DNA polymerase action because DNA it maintains the stability of the single-stranded DNA, ensuring that the DNA polymerase can efficiently replicate the DNA. Much like how a stable workbench helps a craftsperson work efficiently, similar to that, it and also as I already mentioned about the prevention of the secondary structures which it has the ability to form. We can see how these protein come and bind to the uh, single-stranded DNA and prevents it from the formation of any secondary structures which would have been like difficult for the DNA polymerases to work onto. Now, talking about the very important enzyme that is the DNA polymerases, the enzyme that is responsible for synthesizing new DNA strands. Let's think DNA polymerases like an skilled builder who assembles a complex structure with a great precision and speed. Now, the DNA polymerase also comes from the uh, class transferases, and as there are many DNA polymerases involved in the whole process, there are the EC number for all of them are given. Talking about the structure, DNA polymerase has, like all the DNA polymerases, have a common or a general structure in them, which resembles to our right hand with palm, fingers, and thumb. So talking about the palm domain, this domain is involved in the catalysis and is conserved across various families of DNA polymerases. Then there's a finger domain. This domain interacts with incoming nucleotides and the template bases. And then there is thumb domain, which positions duplex DNA and is involved in progressivity and translocation. Depending on their structural differences, there are around five families into which they have been segregated. Talking about family A, they are mainly found in bacteria, especially like the commonly known TAC DNA polymerase, which is used in PCR. Uh, it has a conserved palm domain, but it has unique finger and thumb domain. Where as the family B, which like is mainly present in the eukaryotic polymerases, have a similar palm domain to that of the family A, but unique thumb and finger domains. Talking about the family C, which can be seen in E. coli polymerase 3, there's a multi subunit structure with and a core domain of alpha, beta, and a clamp loading complex. Further, family X is uh, a it's like polymerase beta, like the example of it. Then the structural features is that it's the smaller structure compared to the other families and has a specialized domain for the repair function. Then there is family Y, which has a very unique arrangement suited for the translation synthesis. Talking about the difference that the polymerase of prokaryotes and eukaryotes hold can be clearly seen. The in prokaryotic system there is DNA polymerase 1, 2, 3, 4, and 5. So DNA, talking about one, it's a single polypeptide with distinct domains for polymerization and exonuclease. About two, it's composed of seven subunits, multiple domain architecture it is. And talking about the three, it has 13 subunits, including the core domain and a clamp loading complex. DNA 4, polymerase 4 is a single subunit with unique domain arrangement for the damage tolerance. And polymerase 5 is composed of UMUC monomer and UMUD monomer. Then in eukaryotic DNA polymerase delta is a multiple subunit complex with a distinct domain for polymerization and proofreading activity. Then there's polymerase alpha, which is composed of multiple subunits, including primase and polymerase domain. And there's epsilon, which has multiple domain structure but is specialized for repair. And there's gamma, which has unique adaptation for the DNA replication of the mitochondria. Further, talking about the critical functions that the DNA polymerase carry out is first, DNA polymerase adds new nucleotide to the 3' end of the growing DNA strand. Then there's a leading and a lagging strand synthesis. The leading strand is synthesized continuously, like laying a smooth and a straight path, while the lagging strand, however, is synthesized discontinuously. In short, the segments of Okazaki are formed. Then it has the proofreading and error correction property, which is like the 3' to the 5' exonucleus activity that removes incorrectly paired nucleotides. Then we have the primer removal and replacement, uh, which in prokaryotes is done by DNA polymerase 1, where the RNA primers are removed and replaced with the DNA. Then we also have the processing of the Okazaki fragments, which is done. This is majorly done by removing the RNA primers and filling in the gaps with the DNA ones. Then we have specialized functions which are seen in eukaryotes, is that DNA polymerase alpha is works with the primases and initiates the DNA synthesis. The gamma replicates mitochondrial DNA. As we all know that mitochondria has its own DNA, and the beta is involved in the DNA repair mechanism. Also, talking about the high speed synthesis, polymerases can add nucleotides rapidly with a rate up to 1,000 nucleotides per second in prokaryotes, ensuring efficient replication of the genome. Then we have maintaining replication fidelity, the combined action of polymerization, proofreading, and exonuclease activities that ensures DNA replication proceeds with high accuracy, minimizing errors that could lead to mutation. Due to the activity of proofreading done by DNA polymerase, the accuracy of DNA replication and the chances of having errors is very low as compared to protein synthesis. So we can see that how fastly the DNA polymerase is adding into the nucleotides, and it always works from the 5' end to the 3' end. Lastly, we are talking about DNA ligases, which are the group which are the enzymes that seals the nicks in the DNA and joins the Okazaki fragments. Let's think about DNA ligases as the glue which joins every piece of structure in a secure bonds. Now, these DNA ligases come from the class of ligases, and the EC number is given as there are two DNA ligases. One is ATP dependent, and other is NAD plus dependent. Talking about the structure, the ATP dependent ligases. These ligases are often smaller and consists of two domains. First is the adenylation or the nucleotide binding domain that contains ATP binding sites, and oligonucleotide binding fold domain, which includes twisted beta sheets and an alpha helix, much like a welder's torch that focuses on the flame precisely where it is needed. Then, talking about the NAD plus depend ligases, which is based from the E. coli ligase A. It has a central catalytic core responsible for the main catalytic activity, and the N-terminal domain that helps form ligases AMP intermediate with NAD plus, and C-terminal domain, which includes helix hairpin, helix domain, zinc finger domain, and BRCT domain, which contributes to the structural integrity and function similar to the precision control of the welding machine. Then, talking about the functions that DNA ligases uh, perform is that first joining of the Okazaki fragments. DNA ligases forms phosphodiester bonds between the 3' hydroxyl group of one nucleotide and the 5' phosphate end of the next. And it ensures a continuous DNA strand, and it can be thought as the gluing of two nucleotides with each other. Then there is phosphodiester bond formation. This bond formation is essential for the integrity of the DNA backbone, requiring a free OH group at the 3' end and the phosphate group at the 5' end, much like how precisely this work can be actually done. Then energy utilization is as ATP dependent ligases uses ATP, and the NADP dependent ligases uses NAD plus to power the reaction. The process involves three steps: adenylation, AMP transfer, and phosphodiester bonds. Then we have the ensuring of the DNA integrity, which is done by DNA ligases, that the newly synthesized DNA is continuous and stable, much like a smooth path. Then it also has role in repair and recombination. So right now we are done with the structure and functions of the crude enzymes and protein that is involved in the DNA replication. Talking about the replication fork, uh, the replication fork formed in the prokaryote system is different from the eukaryote system. Now, in prokaryote system, the DNA replication begins at a single origin of the replication on their circular chromosome, creating two replication forks that moves bidirectionally to replicate the entire like chromosome efficiently. uh, in contrast, is the eukaryotic chromosome, which is large and linear, which have multiple origins of replication, resulting in numerous replication forks that form the replication bubble, which eventually merges to ensure the complete and timely replication of their complex genomes. So as we can see here in the picture that we have added, the in the prokaryotic system where the example is given as E. coli, there are two replication forks which are bidirectionally moving, and in case of the eukaryotic chromosomes, there are multiple replication forks which are produced and which will eventually merge to form the DNA, new DNA strand. That's how all the enzymes come together and form the DNA uh, DNA replication complex, and the new that is the daughter DNA segments are formed. These are the links that I have, books and the links that I have used to make this PPT and gather all the information. And that's the wrap for our detailed exploration of the enzymes and the proteins that are involved in the process, ensuring that the DNA replication is proceeded smoothly and accurately. I hope you enjoyed this video, and don't forget to like, and if you have any doubts you could ask me in the comment section. Thank you for watching.